few. That's the vast majority. But some of them are going to be found continuously in the body, and they are split into two categories, latent or persistent, but I will demonstrate for you that it's essentially the same thing. So a latent viral infection is one where the host is infected with the virus, but is asymptomatic, and I give it a little asterisk because it might be something very minor that they're not paying much attention to. And the virus can be undetected. If you look for it, you can find it. But once it's reactivated, sometimes it can be fatal. Now, herpes is not one of these. Yes?
Does this have anything to do with them going into the lysogenic cycle?
Yes.
Okay.
So herpes is this is not fatal, but some can be. Like HPV. Right? In the case of chickenpox, not fatal, but it can show up. So herpes is just a continuous reinfection from cells because it just is already existent and not going away. It is a latent virus. You'll have long periods without any symptoms, and then reactivation. And chickenpox will come back as shingles. In fact, the estimate now, just because of the age of the population, is that 50% of people over 50 will have shingles at some point. So that's a lot of them. Okay, so that's happening. And as I said, HPV, if it's in the cervix, can manufacture cervical cancer. So latent infection, for example, herpes, is because herpes initially infects the epithelia, as we've described. Now, there's like type 1 and type 2 and oral or genitalia. Okay, but the epithelial cells there. Both areas are very sensitive, right? And that just means there's a lot of nerves there, right? A lot of nerve endings. And so the released virus sometimes can travel down the nerve to the basal root ganglion. In other words, it hangs out at the spine. And because these neurons do not regenerate, that's it. You can't get rid of it. And so it can be dormant for years and years. You're going to have it for the rest of your life. Stress will induce the virus to want out, to look for new hosts. And so it will travel down the nerve, but you know, as any given neuron has multiple protrusions, the different dendrites, it might not necessarily go to the exact same spot that the initial infection occurred, but somewhere nearby, okay, because it's the same nerve, okay? And so, if it was lips, it'll come out on the lip, you know, that kind of things, but maybe not in the exact same spot. Yes?
So when you say stress can cause this reactivation, is stress just like increased cortisol levels? Is it like histamine? Like what exactly
No,
is
not
it?
histamine. No.
What exactly is it?
So, I mean, for people who have this, I mean, even a sunburn will do it. Okay, but it can be other like physical and mental stress too.
But is it cortisol levels specifically?
Not specifically.
Why do you call it stress? Why are we calling it stress?
We're calling it stress because it's not optimal conditions for the person. It may or may not be cortisol levels, and I'm going to tell you why. Because as people age, their adrenal function drops all across the board. And so if you look at the cortisol levels, okay, of somebody who is 78 years old, it might seem like within the normal range. If you were 11, this person is actually quite stressed right now. Right? So, like, we can't, like, I'm going to hold off on saying it's cortisol level. Okay? Maybe it's elevated for somebody 78. But it muddies this. All right. So that's late. You got it. Seems like it's gone. Oh, I haven't had an outbreak in years and years and years. Bam. Okay, that's late. Persistent / chronic, there's always a little bit of virus continuously made, So some infected cells are in fact dying and releasing virus, okay, budding or bursting depending. So some virus is always present, and over time those numbers sort of overwhelm the host, and sometimes it is fatal. So this is the measles thing that we're worried about. So with measles, for most people, it's acute. For babies under 6 months, they get like this persistent infection such that little tiny amounts of virus are always being released. And what ends up happening is it starts killing off some of the neurons in the brain, and you get like sclera, which is just a little hard spot in the brain that blocks the nerve signaling, and so it's a sclerosing panencephalitis, and this is fatal. So it's a small fraction of all measles patients, but that's always the fear. Okay, so we try to avoid that. I mean, we know that's now, right over the weekend, there's 700 cases currently in Texas, and just with the rate of infection, you know that one infected person gets 18 people infected. I mean, 700 times 18 is a lot of people, and then it can happen again, depending. Yes?
Can these be acute or latent?
This is what I'm saying. Persistent, no. It's if acute is here, then gone. That's it. Persistent is, it seems like it's acute, it goes down, but there's never really gone. And it keeps putting out more and more virus, and the numbers climb over time until now you have another issue. Yes?
Are persistent viral infections less common because they often result in fatality? Because if you keep killing off a certain type of cell, eventually you don't have any more of those cells anymore.
Well, I'll tell you the reason in like 3 slides. So rubella can cause a progressive encephalitis. So notice it's brain things, both of them. HIV, of course, can cause the demise of your entire immune system. And so we'll remind ourselves about HIV. Okay, so the pictures on the left are actual electron micrograph pictures, but on the right is the cartoon image that we're using. And I asked you to amend it so that you've got little boxes in here to indicate integrates, right? Mm-hmm. Okay. And so then once the virus lands with its GP120 on a T helper cell, on the CD4 of the T helper cell, then it's going to fuse, dump its contents, the capsid self-disassembles, and now the reverse transcriptase copies the RNA into DNA. That is the reverse of transcription. And then during the mitotic event, the nuclear envelope is disassembled, okay? And now the HIV DNA is in the nucleus, and that's where the integrase comes into play to clip the host chromosome and insert the viral DNA. And this is known as provirus. You might remember in the lytic lysogenic cycle we said prophagy. Okay. All right. So it's there till we have the stress, and then it gets transcribed, and then the messenger RNA comes out. Some messages for capsids, some messages for the GP120, okay, which is on the surface. This is an enveloped virus, so it's going to pinch off. So this is going to involve, ER and Golgi and all that. So there you go. And then, yes.
For people who are asymptomatic but carry this or any virus, whatever, does that mean their virus is stuck in the lysogenic phase, or could they also be in the lytic phase?
So let me just continue. I'll play this over here. So I want to finish the HIV story. So upon initial infection, primary infection right here, this here, the blue line indicates the T helper cells, the number of T helper cells. And this is just a sort of average estimate. If you've ever looked at blood work, it's all over the map what we consider normal. I just happen to know platelet count numbers offhand, and the normal range is anywhere between 100,000 and 400,000. That's a huge difference, okay, but that's the normal range. So let's just say that it's normal to have somewhere hovering around 1,000 T helper cell per milliliter blood, okay? It might be a little higher for some people, it might be a little lower, okay, but this is roughly even. Initially, okay, here's your virus, HIV, but HIV preferentially binds to T helper cells, and when it replicates and leaves, it destroys that T helper cell, such that the numbers drop, okay, and the HIV numbers go up. This is the acute HIV syndrome that is typified by seeming kind of like a flu, okay, sort of flu-like symptoms, fever, lethargy, you know, like just kind of flu-like symptoms, and then it seems to subside, right? And it seems like the T helper cells recover some, but you notice they never go back to normal, okay? And now we're looking at many years later. This isn't the same graph anymore now. Many years later, what's happened is that the HIV infection is not acute, in which case the numbers would have come to 0, and that's that. Instead, it's still there, and a little bit are constantly being made and released from infected cells. And as that's happening, we are constantly losing our T helper cells. We don't know this yet, but the T helper cell is basically the director of the immune system. And so this we do need to know. Below 200, when the count of T helper cells goes below 200, that's when clinically it's called AIDS, okay, acquired immunodeficiency syndrome. Because at that point you start getting what are known as constitutional symptoms. Getting things like the Kaposi sarcoma, a type of herpes virus that we've all been exposed to and mounted a response and nothing happened at all, but people with HIV will in fact show signs of it. Toxoplasmosis, that's actually a protist, and it's carried in cat feces, and you've been exposed if you have a cat, and that's not caused you one iota of problems, but if you don't have an immune system, you get toxoplasmosis from changing the cat litter, and it's like, what? So then opportunistic diseases, and you know that is your normal microbiota that now are taking advantage of the fact that you can't keep them in check, okay? So eventually it does lead to death, and look what's been happening to the T helper count the entire time. It's dropping and dropping and dropping and dropping, okay? It is entirely possible to live with HIV and be HIV positive. What has to happen is that you take medications to minimize the amount of viral release, but also these people are told to eat really well, get lots of rest and sleep. Why? To keep the virus happy, out of the stressful level where it wants out, because when it wants out, it's going to destroy all the cells that are carrying it, okay? So then, here the table says, okay, these are latent, you get them, they seem to go away forever, and then here they are again, and here's persistent, okay? And it's like, wait, didn't you just say that HIV is persistent? All right, well, here's our problem, okay? And so this is a big issue in science, and I'll tell you that in grad school, I had an instructor, professor say, you are only as good as your test. And so here's the difference. So here's acute infection. This is most viral diseases are going to be like this. There's a certain number. The eclipse period, it's inside the cell. It's forcing the cell to do all of its biosynthesis, and then it's released. You get a ton out there, and I really like this graph in the sense that you see how huge it is. It is not that slope, steep as it might be for bacteria. It is a boom. It's a slam all at once. But then it gets cleared normally by your immune system. The persistent infection never reaches 0, is continuously being made, okay, until the numbers overwhelm. With the latent, it seems like, well, it's gone, completely gone, and then here it is. Okay. The problem is this: if your test cannot detect below a certain level, and then suddenly here's the virus again, you're going to call it latent. If your test is sensitive enough to always detect some level of virus, you're going to call it persistent. And so I'll give you an example for stuff that, you know, it's not latent, it's an acute infection, but think about the test that we took initially for SARS-CoV-2, okay? first we didn't really have the colored test with the little tabs. You had to get a PCR, right? And so they got your nose swab, they sent it off, and 4 days later or whatever, go online and figure out your yes or no, right? But now we have tests that are mailed to us. They're very, very sensitive. They're called ELISAs, and we're going to do one in lab. Okay? They're so sensitive now that people who feel really well, boy, I've been well for like 10 days, they're still coming up positive, right? Because the sensitivity level is much better. So I'm saying to you, if my 0 is now here, oh, could it be that the latent was down here the whole time, just was undetected? You see what I mean? So I'm not going to get all tripped up and should I call it persistent, should I call it latent? Like, what's your test? And I'll tell you what you're calling it. If your test can't detect it, then you're going to have to say latent. But if your test is like, oh yeah, there it is, there it is, there it is, it keeps getting bigger, that's persistent. We're not going to get tripped up on it because, you know, everything that's in the latent, I'm just going to say they haven't gotten the test sensitive enough to see it. Okay? I mean, if we get like uber sensitivity and I still can't see this thing, okay, we'll call it latent then. Either way, the same thing happens at some later point, here it is again. Okay, that's the main idea. But here, hopefully, Amelia, this is going to answer your question. The extent of the damage that a virus causes is directly related to which cell does it infect. Most viral infections are acute because they're after epithelia. Epithelial tissue is mitotic tissue. You're going to make more anyway. That's your plan. How quickly will you make more? Well, that depends on a lot of stuff. You have ATP to be able to do this. You have the energy, you're eating right, you have all of the building blocks necessary to build a cell. You know it's 4 carbon compounds. Okay, do you have that? If you don't have that available to you, you're not dividing at the same rate as somebody else. Okay, what level of ATP do you have? Well, you know, are you 5? Are you 85? These are different answers, right? So somebody who's young and you know, even older people, if you're in great shape, well rested, eating well, lots of ATP, you'll get over a cold much more readily than somebody whose answers to all of that are no, and not well rested, not eating, right? Stress, all of these others. So, okay, that's most viral infections are after epithelia. If we get a viral infection after a neuron, you're stuck. I am not going to replace that cell. I don't know how. That's over, right? Okay. Hepatocytes. So all of those hepatitis, the real reason we're so concerned, I mean, of course, the liver is such an important organ. I know I say brain, heart, lung to you all the time, but if I had to put a fourth in there, it would be liver. There's so much, 6 different really, really important functions. Hepatocytes, they only divide once a year. So you may as well say you're never going to get over this. Because it doesn't divide all at once. But it's like one cell just divided. That means 365 days from now it'll do it again. A cell here, you know, maybe it's in 2 months' time, and you know, it's not all at once. So basically, you can't ever really get over it. Those viral infections that are after epithelia, those are the acute ones. Okay, so that's something we should recognize. Not all of our tissues and cells divide at all, or divide on the same timetable. Does that make sense? Okay, very good. So I'll just take a second here, just to kind of help you with your whole, like, you know, is this thing alive, or is it? Okay? So with what is this thing? Like, what is this thing? Where did it come from? We have no idea. It's not living, it's not a cell, it has no metabolic machinery, and I know I'm saying it just like that, but what do I mean when I say no metabolic machinery? What does that mean? Yeah, can't make its own ATP. So no enzymes for that, either substrate level or electron transfer chain. Please, come on, that's so complicated. They don't have anything like that, but they can't even ferment. They can't do that either. Okay, they don't have ribosomes. They don't have Golgi. They don't have, you know, rough ER. And it's like, okay, hello, bacteria live just fine without rough ER and Golgi, but they can in fact make ATP. They can in fact break down foods, right? All of those things. Okay, so I just want to make sure you can kind of flesh that out a little bit, because if I say they have no metabolic machinery, and you were hoping that that was what you were going to say, like, gotta be able to flesh that, okay? All right, so genetic material is not always double-stranded DNA. That is the case for every known cell. So whether it's fungi, whether it's protists like algae, okay, or paramecium or amoeba, okay, animal, bacteria, all have that. They don't have that. Now there are in fact some double-stranded DNA viruses. Herpes is such a virus. Smallpox is such a virus, right? Okay, so that it definitely exists. Okay, hepatitis B, okay, but so many others of them. And you might have noticed this as you were looking at those tables. They're mostly RNAs, aren't they? Tons and tons and tons of RNA viruses. Okay, so, but because it relies so heavily on a cell for every last thing, there's the escape hypothesis. That it was inside of a cell, but it took off. Okay, so if we were to look at something like, I'm going to skip the transposon thing, but if we jump to plasmids, what is a plasmid? Small circular piece of DNA. It's not part of the chromosome. It's only 1 to 5%, okay, of bacteria. And so a plasmid has its own origin of replication, just like the viral nucleic acid will, okay? The difference between a plasmid and a virus is that a plasmid can only leave the cell. It never really leaves the cell. It is transferred between cells across cytoplasmic bridges in conjugation, right? It's never like out there loose in the media. I mean, we did that in lab, but you know that's fake, right? Okay, so a virus is kind of like a plasmid that has a protective coat that will let it leave. Okay, does that make sense? All right, that's the thought. So it's like, what if you put capsid genes on a plasmid? Would that then be a virus? And that, I think, would be the beginning of what a virus might be. Okay, but it's not just a capsid. The more important thing would really be the spike. Okay, because if it can't get in, I mean, surviving outside for a while, that's one thing. You gotta get in and basically take over a cell. So that spike is very important too. So, you know, we don't really know where it's from. And the other thing we do know is that because the capsid, so think about size-wise nanometers, so small. So you can only tuck so much nucleic acid in there. And so what happens is that they read their nucleic acids like backwards and forwards and like kind of jump in between. So they're getting a whole lot out of a short segment. So, I mean, years ago when I was a kid, it was like there would be, you know, little puzzles or stories like this where it's like, okay, it's like a sentence, but then it'd be like, you know, over here there'd be a key, like every fourth letter, read it, and it'd be like saying something else entirely, right? That's kind of what they're doing. Like they can read a little piece, bigger piece, okay, and it can actually encode many, many different things off of one small segment. So it's quite efficient, really very efficient. The other thing is that, remember that when we talked about RNA transcription, and we said that the RNA polymerase sits on the promoter, and that was attractive to it, and then it would go ahead and make message. Why is it that nucleic acids from virus are made preferentially over the host cells gene? Why? Viral promoters are very attractive. This is the thing. So, well, why don't we just make ours more attractive? Well, there's reasons we don't turn all our genes on all the time. We're like much more regulated. Okay, and so it's like they're just taking advantage of a system where RNA polymerase doesn't live on the surface of the promoter, just lands, does its thing, and floats off. This is like, oh wait, this is way more attractive, and goes over to the viral promoter. So that's kind of what happens there. And then, like, the last thoughts here are does,
What
yes.
what do you mean by attractive? Like,
Oh, I mean, like, so if you think about nucleic acids, they have the sugar phosphate backbone, so they have some negative charges, but they also have the bases which are hydrophobic. But even on those bases, there's like amine groups, like NH3s and things, and those are also electrophilic, so there's just a little bit of charge attraction.
Okay, so
Yeah.
polarity wise.
It's a polarity
Okay.
thing. Remember a long time ago I said, look, you don't really need to know a lot
Yeah.
of chemistry in here. It's like if you understand water and polarity, and then the only other thing really in that group would be the oxidation reduction. Then you have just covered everything there is to say in most biology centered courses. Okay, so yes.
So everything about viruses that appear just screams like unstable. Like,
Oh yeah, no, they're completely unstable. Yeah.
So is the only thing that's really holding them together the capsid? Is that pretty much it?
Well, so they only need that capsid for the moment, right?
Uh-huh.
Just as a delivery
Just to
vehicle
keep them together to deliver
to
something.
deliver it. And then once they're inside the cell, well then a cell is a very stable thing. So they're fine inside the cell.
And they
But
use
you know, you
the same
squirt them or they're on the desk. I mean, they only have so long. They're at the bottom of my table of who's resistant and who's not. They have very, very little going for them. Okay? They cannot do homeostasis, right? Keep themselves from drying out right here. Yeah, they're weird. They're really weird. But so we also know this. Virus absolutely introduce genes between cells in bacteria. This is called transduction. They're super sloppy when they leave. They don't even care. So they trick this cell into becoming a virus-making factory, okay. But when it leaves, some of them, by mistake, mispackaging, take up some bacterial chromosome. Okay, and when they leave, they'll go infect another cell and they'll squirt in not virus genes, but guy next door gene. So this cell isn't going to become a virus-making factory, but you know what it is? It just picked up a new gene. That's transduction. It is not mutation. Mutation is different, right? So either I'm changing a base, right? Okay, so, you know, I mean, you could say yes in terms of insertion. Okay, like a frame shift just happened here, but it usually is just an addition of a gene. You know, it's like it's picking something extra up. Yes.
The speeding up is only due to bacteria, right?
Yes, this here, right? Okay. On this slide. And then we go here. And this is what happens with influenza. So influenza, you know, is after epithelial tissue of the respiratory system, but its host range is vertebrates. So I have an image here of a duck and a person. They both have the flu. The duck has a certain kind of influenza, let's say a H1N1. Let's just say that's the one that the duck has. And the person has H2N2. Okay, so they're slightly the same, but slightly different. Okay. And the pig, who is nearby both of them, gets sick from both. Okay, gets influenza from the duck and gets influenza from the person. Inside the infected epithelial cells of the pig, the virus is really sloppy. And when it packages itself up and takes off, it's now a hybrid, such that the released thing maybe has H1N2 mixed up. This is called antigenic shift. Antigenic shift is why we have a really hard time keeping up with the flu. That and the fact it has replicase and there's 8 strands of RNA to mess up, right? Okay? So this new flu may well get the person sick all over again if they don't quite recognize it. Okay? So this is what's happening. And so, wait, so this is now changed? Yes. Okay, so you maybe are getting some duck genes. You know, I can't say you're not. What if you're getting it from like another person? Are you getting like a slight version of theirs? You know, I can't say that isn't happening. I can't. So are we speeding up evolution? Evolution being changing the alleles, the frequency of alleles in a population over time? I can't say that's not happening. We don't know. We got a big fat question mark on it. Okay, we don't know, but we do know that this happens all the time. Antigenic shift. Okay? And then lastly, viruses and cancer. And I think I drove this out for you last time, didn't I? With HPV.
You talked about it, how HPV causes certain.
But I did not describe how. Okay. Okay, so I need to do something just so that I can, this is the cell cycle. Okay, you're a eukaryote, not a bacteria, you know that part. Okay, so you're not doing binary fission, you have a cell cycle. Okay, and so in a cell cycle, a cell divides into two, and then they're smaller, right? So the new cells are smaller. Okay, so now they're in G1, which stands for gap 1, and all it means is that that's a section of time where the cell is just growing and starting to express all the genes that are common to that cell type. So if this is a cell from epithelia, then it's going to start expressing all of the proteins on the surface and within it that are unique to epithelia. If this is a cell from a hepatocyte, then it will start expressing and making all the proteins associated with the hepatocyte. Yes? Okay, so that's development essentially. So gene expression, which basically is like, get me the proteins I need, basically. And it's also, I'm going to write it like this, amassing ATP, because you can't really, it's in constant use, you can't really amass ATP, but that's what they try to do. Okay, once they do so, they go through a checkpoint right here. Okay, the cell decides whether to continue. This is a checkpoint. And at this checkpoint, the question is enough ATP. Is there enough ATP? Why? Because the next thing is S, which is the replication of DNA. It stands for DNA synthesis. Okay, synthesis of DNA, and that's a big deal. Right? Synthesis of DNA, copying your entire genome. You have, 23 pairs. It's got to be copied top to bottom. All of the stuff that happens. It takes a lot of energy to do that. Okay, fine. The DNA is now replicated, and then we go on into G2. But right here, there is a checkpoint. Oh no, no. Great. And at checkpoint 2, so the DNA has been copied, and now we have to subfractionate the organelles. Because if you're about to divide into 2, well then, I need some rough ER also, right? And Golgi, and all of that stuff is going to be like separated into 2, okay? And amass ATP. And so before we go further, the questions are all the DNA copied? Organelles split up, and enough ATP. All right, and then we start the mitosis process, which, I told you this, I'm sure many times, that I used to work with B cells for many years before I went to graduate school. And they're like the fastest dividing cells we have, 24 to 26 hours. They spend 2 hours in mitosis. So really, this should be a tiny fraction on this circle, okay? the S phase takes a long time. And for many cells, they'll be stuck at G1 for a really long time, especially if it's a very specialized cell with lots of specific things that they need to make and secrete and all that. Okay, they might be stuck there for a really long time. So, and you know, some cells like just pop out altogether and go to G0, right? Okay, cardiac muscle, neurons. Okay. Can't get that. All right, but during the mitosis process, you've got your prophase, that's where your nuclear envelope breaks apart, you know, and I know it's early, middle, and late prophase, but then it's metaphase, and in metaphase, what happens is, all of your sister chromatid have to be lined up on the equator. Okay, there is no equator, just like there isn't one on the Earth either. They just mythical center. All right, that's a checkpoint. So I'll put it here, metaphase. Is checkpoint 3, and that's a really important one, which is sister chromatids align. Because from this point forward, the cell pulls apart. Okay, so if you did it wrong and one side gets more DNA than the other, then we're messed up. Yes? Okay. So we have these checkpoints, and so you know the ATP question is actually quite large. If the answer to any of these is no, we pause. We can pause at G1 till we get enough ATP. If at checkpoint 2, did we copy the DNA correctly? If the answer to that is no, the cell undergoes what's known as apoptosis, programmed cell death. It will just kill itself. Nope, not going forward. Okay. Checkpoint 3. Are we all at the alignment? Okay. It gives itself a little chance to get there, and if not, it goes through apoptosis, programmed cell death. It does not go forward. Okay. What do you think is in charge of all of this? The secret answer in this class, proteins. Of course. Specialized proteins known as cyclins. Right? Very, very important proteins in this process. Okay? Cyclins are proteins. Their information is where?
On the DNA.
Yeah, it's on the DNA. And so, let's just say that this is the DNA for the cyclin gene. For one of them, because there's lots of cyclins. But, you know, okay. Let's just say here's one gene for the cyclin. And let's just say that you have an HPV. Okay? HPV, naked DNA virus. It's after basal epithelia. During mitosis, it gets inside the nucleus, right? Okay? And so it will wind up destroying that cell. But sometimes it incorporates into the DNA. And if it were to do so, here, let's just say, kind of in the middle of this cyclin gene, so let me just make it a little bit longer. And what have I just done to the cyclin gene? That's like having a big insertion, isn't it? What does that do? Is that a good mutation?
No.
Not at all. That's the worst kind. Nonsense and frameshift, those are the worst kind. So what just happened to my cyclin? Is it functional? No. Do I have enough ATP? Keep going. What about, did you do everything right? Keep going. Everybody, who cares? Just keep going. And so pretty soon you're looking at a cell whose cycle is just this, S and M. Okay. Copy the DNA and divide. Copy the DNA and divide. That's it. Dividing out of control, that's cancer. That is the generic terminology for cancer. Cells that divide out of control. This is how a virus can cause cancer, by disrupting the gene for a cyclin. okay? Let's be clear on the wording here. I don't want to like minus a half you, okay? So let's just make sure that we're saying we are disrupting the gene for the cyclin. The cyclin is a protein. Information for it is on the DNA. This is what gets disrupted. Do not tell me that the virus inserts its DNA into the protein, because that's what? No. Proteins don't have DNA stuck in the middle of it. That makes no sense. Okay? All right. So this business here, where it just copies the DNA, divides, what's missing here is this part, where the genes are being expressed, the specific proteins that are like that tissue. And over time, cells that are cancerous, they stop looking like the cells that they started from. Okay? And the further away it is, then those are the stages. If you're at stage 1 cancer, okay, this is happening, it's dividing out of control, but still really looks like the cell it started in. Stage 2, ooh, less so. By the time it's stage 4, this is where they're like, if you hear about somebody they just found stage 4 cancer, they're scrambling, trying to figure out where the heck did this thing start, like, because it's a cell without any characteristics, any defining characteristics, other than just being a eukaryote. Okay, nothing. The only time in your life where this is normal was back here, okay, where that cell divided into 2 and divided into 4 and all of the other stuff and became a little ball of teeny tiny cells that never really did grow and aren't really look like anything, and that's totally fine, okay? But by the time you know that not doing it nice, you know what I mean, okay? When this thing becomes this, that's all over now. Now we're not doing that anymore. So if they take somebody's blood and they find things like alpha fetal protein and you are not pregnant, this is an issue. You should, what? Why is that happening? You should not be expressing that. That indicates to me you have cells dividing out of control. Okay? Most viral infections, that's not what they're gonna do. We saw our tables, they're mostly RNA. That's not their plan, climb into your DNA. That's not their plan. The DNA viruses, the retroviruses, those that can become RNAs that become DNA, like HIV, that might be their plan. That might end up happening. That might do this. That's not always going to be the case. We have a ton of DNA. Okay? A lot of it doesn't speak anymore, which means it doesn't get expressed. And so if a gene from a virus is inserted into some of my DNA, but I wasn't going to say anything there anyway, you might not even know. Okay? So, okay, yes.
So are the cyclin proteins, are they in charge of these checkpoints?
Yes.
Okay. That's all I was.
Yep. Mm-hmm. Yes.
What is the likelihood that it actually attaches to the cyclin protein?
Well, unfortunately, so there's like 120 different versions of HPV, and there's like 2 or 3 that cause cervical cancer. Most HPVs cause warts. Just,
So it's
benign
attracted to
warts.
this section specifically.
Well, that's not well understood.
Okay.
That is not well understood. Nor where it is that HIV has decided to insert itself. Not well understood. Seems to be almost anywhere. Yes.
So does cyclin is also the protein that activates the
Apoptosis? No, that's a different set of proteins. Okay, good here. Yeah. All right, so. So that's pretty much all I wanted to say for virus 3. Okay, so let's spend the rest of the period